All right, we're about to go microscopic to understand something that is, well, truly massive. What if I told you that everything about you, the bacteria in the soil, every single living thing on this planet, is all powered by the exact same tiny, intricate set of molecular machines? Let's dive in and take a closer look. So, here's a question for you. What do you, a mushroom, and the very air we breathe all have in common? I know, it sounds like the start of a really weird joke, doesn't it? But this question, it gets right to the very heart of biology. So how are all these things connected? Go on, think about it for a second. Any ideas? Well, the answer is actually incredible in its simplicity. We are all, and I mean all of us, built from and completely dependent on the exact same set of molecular building blocks. It is the great unifying principle for all of life on Earth. So it's time to meet these master molecules that make, well, everything possible. It's honestly amazing when you think about it. All of the staggering complexity of life, it all boils down to just four main types of large molecules, or as scientists call them, macromolecules. You can think of them like the ultimate Lego bricks that build everything from a tiny virus to a gigantic blue whale. So let's meet the core four. First up, carbohydrates. They're basically the gasoline for your cells. Quick, easy energy. Then you've got lipids, things like fats and oils. They're the battery packs, storing energy for the long haul and forming the boundaries of our cells. Next, nucleic acids. You know these ones, DNA and RNA. They are the architectural blueprints holding all the instructions to build and run you. And then there are the proteins. These guys are the real workers. They're the enzymes, the transporters, the structural beams. They do just about everything. And what's really fascinating is the simple principle behind all this complexity. Each of these big molecules is what we call a polymer, which is really just a long train. And each train is built from smaller, repeating rail cars called monomers. For carbs, the cars are simple sugars. For proteins, they're amino acids. It's this simple pattern, just linking these little monomers into huge polymers, that lets nature build such immense complexity from a surprisingly small set of parts. Okay, so for the rest of our time together, we're going to zero in on that last group, the proteins, because they have one of the most dynamic and absolutely critical jobs in your body. They act as enzymes. So what in the world is an enzyme? Well, think of it as a biological accelerator. It can take a chemical reaction that might take years to happen on its own and make it happen in a fraction of a second. Seriously, without enzymes, the reactions that power your body would slow to a crawl, and life would be totally impossible. Now, here's where it gets really cool. An enzyme's power comes entirely from its unique 3D shape. It's folded up into this very specific, intricate structure, and somewhere on that structure is a perfectly shaped little pocket. This is the active site. This is the business end of the enzyme. And the process is just so elegant. First, the target molecule, which we call the substrate, it finds the enzyme. Then it binds perfectly into that active site, kind of like a key fitting into a lock. This connection creates the perfect conditions for the chemical reaction to happen at lightning speed. And here's the best part. Once the reaction's done, the enzyme just lets go of the new products and is immediately ready for the next job. It's the ultimate reusable tool, doing its task over and over and over again. So how exactly are enzymes speeding things up so much? Well, every single chemical reaction needs a little push to get started. Think of it like an energy hill it needs to climb first. Scientists have a name for this. It's called the activation energy. And here is the real magic. Enzymes don't add more energy to the system. Instead, they just dramatically lower that energy hill. They make the reaction so much easier to start that it can happen millions of times faster. It's the difference between trying to push a giant boulder up a mountain versus just giving it a little nudge to get it rolling down a small hill. Okay, so now that we know how these incredible little machines work, the next logical question has to be, how do you stop them? How do you throw a wrench in the gears and sabotage the machine? Well, you can stop an enzyme with a different kind of molecule called an inhibitor. You can really just think of it as a wrench in the works. It's a molecule designed to get in the way and stop the enzyme from doing its job. And, inhibitors can be pretty clever about how they pull this off. There are two main strategies. First up, you've got your competitive inhibitor. This molecule looks so much like the real substrate that it can actually sneak into the active site and just sit there, blocking it. It's like someone parking their car in your reserved spot. Then there's the non-competitive inhibitor. This one takes a totally different approach. It binds to another part of the enzyme entirely, and in doing so, it actually changes the enzyme shape. This warps the active site, so the real key just doesn't fit the lock anymore. And listen, This isn't just some abstract theory. This is literally a matter of life and death. Carbon monoxide is a permanent competitive inhibitor. It gets into the active site of a crucial protein in your red blood cells, gets stuck, and physically stops oxygen from binding. That is why it is so incredibly toxic. But it's not just these blocker molecules that can stop an enzyme. Remember how absolutely critical that specific 3D shape is? Well, it turns out it's surprisingly fragile. Extreme conditions, like really high heat or a big swing in acidity, what scientists call pH, can cause that whole intricate structure to completely unravel. This process is called denaturation. And a denatured enzyme is a useless enzyme. Just think about cooking an egg. The heat changes those clear, liquid egg white proteins into a solid white mass. You can cool the egg back down, but it is never going back to being a clear liquid. The change is permanent. The machine is broken for good. So we've seen how these machines can be broken at the molecular level, but now let's zoom all the way out. What happens when a single enzyme isn't just important for a cell, but actually holds the key to feeding the entire planet? Just take a deep breath right now. About 78% of the air you just inhaled was nitrogen gas. Nitrogen is a vital piece of our proteins and our DNA. We absolutely need it to live. But we have a huge problem. You see, the two nitrogen atoms in that gas are held together by an incredibly strong triple bond. It's one of the toughest chemical bonds in all of nature. And for most living things, it is completely unbreakable. We are literally swimming in an ocean of nitrogen, but we're starving for it because we just can't access it. Or we would be starving if it weren't for some very special bacteria. These microbes have a secret weapon, a super enzyme called nitrogenase. It's one of the only things in nature powerful enough to actually break that triple bond. It fixes the nitrogen, turning it into usable ammonia. This one enzyme is the gateway for pretty much all nitrogen to enter the global food web. For centuries, this natural process was the bottleneck for all of agriculture. But then, in the early 20th century, scientists figured out the Haber-Bosch process, an industrial reaction that basically mimics what nitrogenase does. This invention led to synthetic fertilizers, and it completely revolutionized farming, allowing us to grow enough food to support a global population that has absolutely exploded ever since. But this incredible new power came with a serious downside. It turns out plants only absorb about half the fertilizer we put on them. The rest just washes away into our rivers and oceans. And this flood of nutrients triggers these massive algae blooms that then choke the water, sucking out all the oxygen and creating vast aquatic dead zones where nothing can live. It's a powerful reminder that even our most brilliant solutions can have complex, unintended consequences. So we've seen it over and over again. For a protein, shape is destiny. Its function, its power, its entire purpose, it all comes down to that specific 3D structure. Which leaves us with one final, fascinating question. What happens when a protein folds itself into the wrong shape? Well, the consequences of those simple misfolds, everything from devastating diseases to the very nature of life itself, that's a story for another time.